Good morning. Question number 63. Jharkhand eligibility test life science detailed solution. Okay, let's see the question. The question is a cloning experiment uses directional cloning with two different restriction enzymes. The main advantage is increased options are increased plasmid copy number, prevention of vector self-ligation and control of insert orientation, increased mutation rate and improved transformation efficiency. Okay, let's see. This is the question, and these are the terms you should be uh, knowing about previously. Okay, the cloning experiment is any group of cells in which the genes are same, in isn't it? The DNA or chromosome are the same is known as clones, and these the production of these are is known as cloning experiment. Restriction enzymes are also known as molecular seizures. These are used for the cutting of DNA, isn't it? Plasmid is the extra chromosomal material that is present in an organism to provide its extra ability despite its normal genetic material. Copy number is the average number of molecule of plasmid or gene per genome contained in a cell or an organism. Vector is the plasmid, phage, virus or bacterium. Anything that carries your interested DNA into the host is known as vector. Ligation is joining of DNA fragments to produce a single DNA molecule. Mutant is any cell or organism other than the normal genetic material is known as mutant. Transformation is the introduction of DNA into the cells of an organism by method other than sexual crossing, isn't it? Transformation means the normal cell has been transformed or has been mutated to grow into something, some new organism, isn't it? Some new species or per se. Let's see. So, talking about molecular cloning, what is molecular clone and what is a plasmid or cloning vector? We will see about this. Molecular clone is the nucleic acid fragment of interest that has been transferred to a cloning vector, isn't it? And the molecular clone is genetically similar to the normal organism or normal cell, isn't it? Just to make a copy of a cell is known as molecular clone. So cloning is uh, when a cloning is done, then a cloning vector is cleaved with restriction endonuclease. Say, for example, a human chromosome is to be a gene of, let's say, insulin has to be created in the bacterium, isn't it? For its production, the insulin gene, which has been shown in red, isn't it? The gene of interest shown in red will be cut to attach itself to the plasmid, isn't it? So it is the gene of interest that is, let's say, that is red is fragmented or cleaved or cut by the help of restriction endonuclease, and that restriction that fragment or gene of interest is attached to the plasmid vector, isn't it? And this vector is ligate or attached to the plasmid vector, isn't it? And then the DNA or the newly inserted gene in along with the vector is introduced into the host. So this vector has let's see the example of this vector which has been introduced into the host. So this is a example of PBR322, and it has two ampicillin resistant, one ampicillin resistant genes, one tetracycline resistant genes, and one origin of replication, isn't it? So here is another, the same, having three different recognition sites, three different restriction enzyme site, isn't it? See, this is a figure of PBR322. It has an origin of replication from which the replication takes place, and it has four restriction sites: PVO2, BAM-H1, ECO-R1, and PST1. And inside ampicillin resistant gene there is a recognition restriction site, and inside tetracycline gene there is a restriction site. So again, revising that molecular workflow, we take the in step one, we isolate the inserted DNA from a gene of interest from that organism that is ligated with the plasmid vector with the help of restriction enzyme, isn't it? And that is again inserted into or transformed into the host cell, and that is again produced for large scale production and verification and restriction analysis, so that to check whether the cloning has taken place successfully or not. So in question number 61 we have seen that naturally occurring cloning in agrobacterium and TI plasmid was transferred into the plant cell, isn't it? Do see that question. So question was, a cloning experiment uses directional cloning with two different restriction enzymes. The main advantage is, isn't it? So if we use two different restriction enzyme, in case instead of one, what does it help us with, isn't it? So see here, if let's say we are using a plasmid vector that has three restriction enzyme, BAM H1, BAM H1 and ECO R1, isn't it? If we use two restriction enzyme, that is one at one end the ECO R1 is cuts and at another end BAM H1 cut, so it has produced sticky ends, isn't it? Non-sticky end, that is ECO R1 and BAM H1, which will not match and will not stick itself to circularize the plasmid vector, isn't it? And that way it will not be a disturbance in the workflow of the cloning in the cloning experiment, isn't it? If we let's say if there were if we would have used the only BAM H1 site, isn't it? So the ECO R1 site will not play a role in it, and the BAM H1 will lead to both production of both sticky ends on both the ends, and the plasmid vector with self-ligate will self-ligate itself, and that will prevent the workflow, prevent that gene of interest to get incorporated into this plasmid vector, isn't it? Again, option number one was increased plasmid copy number, isn't it? So option number, plasmid copy number is totally dependent on origin of replication, isn't it? You can see here plasmid copy number depends on origin of replication and the vector which has been taken place. So option number C is standard cloning. Option number C is increased mutation rate. See, in general cloning experiment, generally the mutation rate is remain mutation rate does not takes place, isn't it? It has nothing to do with this cloning experiment, and it has nothing also to do with the restriction enzyme. And option number D is transformation efficiency, has nothing to do with restriction enzyme. Transformation efficiency depends on host cell competency and delivery method, isn't it? So here I have given you the AI produced figure. You can go through these. Option A, high copy number dependence on origin of replication, that is option B, production of self-ligating or self attaching plasmid vector, why it happens and why it does not happens. This is the option number B. You can see here, in case of production of ECO R1 and BAM H1 site, it produces it does not match each other, and the ligation does not takes place. Option number C is standard cloning strategies. So standard cloning strategies is these you can see Gibson assembly, gateway cloning system and transfer cloning system. These are you can see for reference purposes. And option number D was improved transformation efficiency. Transformation efficiency is for it, I have given this one. Transformation efficiency depends on the method in which the transformation takes place. Can it be heat shock method or electrocompetency or chemical transformation or electroporation with the help of gene gun or whatnot. Today whatnot we have we have many processes during which can be help to insert the vector into the host cell, isn't it? So thank you for having me.